This IB revision video for module enzymes and metabolism. And remember, this video is to support you. It is not about all the content that needs to be covered, so you will need to look at your own content from lesson. Introduce then, enzymes are all what we call globular proteins, and you should study about what a globular protein is in that appropriate part of the biological molecules module. Enzymes have a very precise tertiary structure, which again you'll have studied during proteins, which gives it that very precise 3D shape. And that is due to the order of amino acids and then the bonding involved, 3 types of bonding. All enzymes are water soluble, so they'll interact. All enzymes act as biological catalysts. All enzymes are not chemically altered in the reactions. They are specific, in other words, they will catalyze only one substrate only. If they have a complementary shape to the active site, they're affected by pH and temperature, and the active site is often only just a small part of the protein, normally about 10 amino acids in size. The shape is maintained overall by the tertiary structure of the protein, and it's vital and has very specific function and can be altered, as we'll look at later, about glycation. Enzymes are biological catalysts. Effectively, they lower the activation energy, so they do the same as a catalyst does, but they are much more specific, and that means you get less unwanted byproducts, whereas catalysts are often, which used in industry, are normally metals, and they are a lot slower in comparison and will produce less specific products, which explains why enzymes are much more used these days in industry. So you need to understand that metabolism is the sum of all chemical reactions in an organism, and metabolism is enzyme driven. There are 2 main reactions which drive metabolism: condensation and hydrolysis. As you know, condensation is used to make new bonds, hydrolysis is used to break bonds. And metabolism is part of a much more complicated series of reactions. One cell can have over 1,000 enzymes because reactions like respiration is actually a long series of chains of reactions, as you'll find out in a couple of videos. If you look at this, you need to know that reactions in metabolism are enzyme driven, and they're often cycles. And what happens is different enzymes are needed at different stages. So for example, the enzyme at one stage may be create a substrate, turn a substrate into a product which drives the enzyme at the next stage. There are 2 types of metabolism really that you need to know about: catabolism and anabolism. So catabolism is where we break things down, so hydrolysis reactions, and just put my pointer on for a second. So that would be that way. So we're breaking carbohydrates down in respiration into water and carbon dioxide, for example. And anabolism would be your condensation reactions, which are turning things like amino acids that you've eaten and been digested into amino acids into proteins for growth. So catabolic reactions break bigger molecules down into small ones. Anabolic reactions break smaller molecules, build small molecules, into bigger ones. Enzymes are specific because they have an active site, and that's the main part of the reaction where things happen. So the active site is a complementary shape to the substrate, which is the molecule that's involved in the reaction. So for example, if you're breaking glucose down, glucose is that substrate. There is a theory called the lock and key theory, and this shows you that the substrate, which you picture here, fits nicely into the active site. We say they have a complementary shape, and when they come together they form something called the enzyme substrate complex. That then puts pressure on the bonds in the substrate, potentially breaking them, as in this example. The subproducts no longer fit and are released back, and the enzyme can be reused over and over again. Collisions can obviously be affected by a number of things, but mainly temperature. And here you can see some substrates moving around, the orange ones being the substrates. This is the enzyme here in green, and when they collide, they will form enzyme substrate complexes. So all particles in a liquid will move more at higher temperatures. Substrates have to be aligned correctly to get a successful collision. Some enzymes will attract the substrate and adjust their orientation to help them combine and form enzyme substrate complexes, but this only works over shorter distances. So basically, some reactions, the enzyme might be trapped in place, as you can see in this diagram at the bottom here. Some reactions, the enzyme might be moving around. For example, DNA polymerase moves along the DNA molecule during replication. Sometimes they may both be free. And therefore it relies a lot on collisions and the energy as a result. Some substrates, as I said, are large and the enzyme might move along them, or sometimes the enzymes are immobilized. An example of this one at the bottom would be the digestive enzyme maltase, which is embedded in the membrane of the ileum. Then we have something called induced fit hypothesis. So whereas we've got the lock and key theory, which is the idea that the substrate is complementary to the active site, the induced fit hypothesis just takes that a little bit further and suggests that the enzyme and the active site here may just accommodate itself to fit the correct substrate. So we know that we think that the molecules change shape slightly as it collides with the substrate, and the active site will fit more tightly around the substrate. And this is called the enzyme substrate complex. And one thing that we think is if there is a charge on the active site, so let's say here there is a plus charge, there's probably a minus charge here to attract the two together. And the change of shape then puts stress on the substrate molecule, destabilizing it and lowering the activation. So if you're asked to describe it, you could say the substrate motion is random until near the enzyme, when close chemical properties activate the active site, attracting the substrate, i.e. using opposite charges. This forms an enzyme substrate complex, and bond angles and lengths change fitting together, and change to substrates make easier for bonds to break, converting them into products. Then the products have changed shape, they no longer fit, so the products detach and return. The active site returns to being empty so it can attract the next substrate. So this specificity is really important. This is what makes enzymes specific. They'll only break down mainly one product. So for example, glucokinase will only ever bind to the substrate glucose, but some are less specific. So there's something called hexokinase, which will bind to any hexose sugar, and proteases will virtually digest most polypeptides. So whilst you may have heard of specific ones such as pepsin, actually they're quite generic as protein digesters. We use enzymes for digestion, okay? We can use them in intracellular digestion. So here you can see a white blood cell, got the bacterial cell, it's taken in via endocytosis into a vesicle, and then they fuse with the lysozymes, releasing hydrolytic enzymes to digest the bacteria. Digestion, however, on a larger scale for all organisms is actually extracellular, i.e. we release enzymes into spaces such as the small intestines. Humans release it into the digestive system, but unicellular organisms and fungi will actually complete endocytosis where they release it into the atmosphere, into the environment, sorry. So here you can see the fungus releasing enzymes into the soil, digesting the food out here, and then the food is reabsorbed in via diffusion. So the enzymes are secreted outside the cell, converting macromonomers to monomers, and through hydrolysis. Remember, digestion is always hydrolysis reaction, and then they can be reabsorbed across the plasma membrane. But really, really important that you remember hydrolysis is always the reaction for digestion. So we also need to look at how do we actually measure enzyme activity. So enzyme activity is important if we want to calculate the rate. The enzyme, as we said, the active site is a very small area. It relies on weak interactions, including between amino acids, including hydrogen bonds, to keep that active site perfectly the right shape. Impacts on these can mean it's no longer complementary and the enzyme is denatured. So we know that molecules are constantly moving. We know, therefore, there's increased collisions as the temperature goes up. So heating it from cold to warm will give more kinetic energy and the molecules will move faster, increasing the number and force of collisions. Enzymes can only catalyze if they come into contact, so this random movement, increased temperatures means more collisions and more enzyme substrate complexes are. This graph should be very familiar with you. So at low temperatures, you have low energy, low kinetic energy of substrates and enzymes, low amounts of collisions, very few enzyme substrate complexes. However, as the temperature increases, the enzyme substrate complex, the energy becomes more available, more collisions, more enzyme substrate complexes. We get to the optimum temperature here, which is where you form most enzyme substrate complexes, and then very quickly afterwards the rate drops because the enzyme is denatured, and that is because the heat energy breaks down the bonds, and the hydrogen bonds particularly break down, which unravel the active site, changing the structure, so it's no longer complementary to the substrate, and therefore the enzyme substrate complex breaks down. pH also has a similar effect. To understand how pH affects the rate of enzyme reactions, we have to understand what pH is. And basically for biology, we just need to understand it's a measurement of hydrogen concentration. And the more acidic something is, the more hydrogens there are present. Now, by having those extra hydrogens, that can greatly therefore affect the rate, hydrogen bonds in the enzyme, which remember are one of the main things that hold the active site in its specific shape. If you have more hydrogen bonds, then it's going to cause different interactions. So here you can see the alpha helix shape of a protein is held together by hydrogen bonds. But if you start adding these hydrogens here, which are from an acid, it can cause different interactions to occur. The hydrogen bonds that would normally hold the structure of the protein together will interact with the acid hydrogens, ions, and then that will alter interfere with them. So altering the hydrogen concentration around the enzymes can alter the bonding, which therefore has a knock-on effect on the tertiary structure and on the active site, meaning it's no longer complementary to the substrate and therefore can denature. It also has a direct effect on the active site because if you think about what we said earlier at the induce fit hypothesis, this relies on charges between groups of amino acids to help them bind to the substrate, and therefore this can directly change those charges and how they interact and prevent the enzyme substrate complexes from being formed. We also need to know what will affect the rate of enzyme activity, maybe not through being denatured, and one of them is altering the substrate concentration. So the substrate we can increase and keep the enzyme concentration the same. And what you'll see happens is the rate will increase as you add more and more substrates until a certain point, and at that point you have no spare active sites available. So the enzyme substrate complexes are fully occupied and you can't get any more because you don't have any more enzymes, you have no spare active sites. So the level will just level off, and at this point the number of active sites is your limiting factor. So here's another picture of it. Here you see if you add more substrates, you've got spare active sites. So if you add more substrates, it will go quicker. But up here, it doesn't matter how many more substrates we add, there's no spare active sites. We can calculate the rate of reaction by doing either average substrate used up divided by time or the amount of product formed divided by time. Use that to calculate the rate of reactions. Now, all reactions will go fastest at the start when there are more substrates and enzyme available, and then the substrate levels will start to drop and the enzyme and the reaction will hit a maximum. So we normally calculate the initial rate of reaction. We can do this by using a gradient and this is a quick way of showing you how to do it. This needs to be covered in more detail. You need to practice this, but really quickly, you have your graph. Imagine the line continues from that graph. I wanted to calculate the rate of reaction at the start. I would draw a tangent, and then I would calculate the initial rate of reaction by doing the volume divided by the time. difference in Y divided by the difference in X. And then the final thing we look at is metabolism. And before, there are two different main types of metabolism: catabolic reactions and anabolic reactions. Catabolic is when you break things down using hydrolysis reactions, so it's digestion of food, cellular respiration and decomposition, whereas anabolism is building things up, so turning amino acids into protein, building nucleotides into DNA, or the synthesis of complex carbohydrates such as starch and cellulose from alpha glucose or beta glucose. Metabolism is enzyme driven. As we said before, it can be a series of reactions with multiple steps. So the enzyme will create the substrate of one reaction into the product. That product is then the substrate for the next enzyme, and so on. So you can see that here, this creates a product, which is the enzyme as that enzyme, which creates a product, which goes to the next enzyme. And that can be a chain reaction, such as glycogen here, which you just go down a straightforward linear chain, or it could be a cycle, such as the ornithine cycle or the Krebs cycle. Inhibitors are important in metabolism because they can control the rate of an enzyme reaction. And there are two types of inhibitors which slow down the enzyme activity. The first type is a competitive inhibitor. And here the blue or purpley colour is the substrate and the orange colour is the inhibitor. So the inhibitor basically will have a shape that can block the active site. So if there's lots of inhibitors around, less enzyme substrate complexes can form. And here you can see if this is where there's no inhibitor, the rate of reaction is quick and then levels off. The inhibitor will slow it, but if you add more and more substrate, eventually there'll be so much substrate that it will almost certainly get into the active site over the inhibitor. The second type is a non-competitive inhibitor, and this attaches to the enzyme somewhere other than the active site, and this is called the allosteric site. And basically when it binds, it alters the tertiary structure of the enzyme, altering the shape of the active site. So you can see here the active site has changed shape, and the enzyme's substrate is no longer complementary to the active site. So therefore, without a competitive inhibitor, you have that reaction. With a competitive inhibitor, it doesn't matter how much substrate you add, the active site is no longer complementary, and it will slow the rate of reaction. Allosteric site is a second binding site changing the overall shape of the enzyme. They have evolved to allow the control of enzyme activity. So they can be reversed, and they can turn an enzyme on or off within a living organism. And therefore, you need to just know an inhibitor is any substance that can slow down the rate of an enzyme control reaction in some way. Often competitive inhibitors are reversible, and not always, but the non-competitive inhibitors are sometimes irreversible. I would say ignore that line there saying that they are normally permanent. They can be permanent as a result. You also need to understand this idea that the substrate can be turned into the product in the next reaction, and the idea of end product inhibition in metabolism. So this is the idea that if you have a substrate, enzyme A converts it into a product which is called intermediate 1. That is the substrate for enzyme B, which converts it into the next product, which is a substrate substrate for enzyme C, which converts that into a product. That's a substrate then for enzyme D, and it creates an end product. To control the amount, we use negative feedback because we don't want tons and tons and tons of this end product. So what will happen is the end product actually inhibits enzyme A. So if you have lots of end product, it will actually inhibit enzyme A, which will slow that reaction, which will slow that reaction, which will slow that reaction, dropping the amount of end product. That then will mean that enzyme A is no longer inhibited, so you get more of that, more of that, more of that, and more end product, and that will then inhibit again. So this is called negative feedback, a process which reverses a change in the body. In this case, it's reversing the amount of enzyme product being formed. Too much product will cause increased inhibition, so the pathway is effectively switched off. Too little product will minimize inhibition, so the pathway is switched on. To finish, you need to know there are some irreversible binding of inhibitors. Mercury, which is a poison, is a non-specific inhibitor as it binds to a wide range of enzymes and is irreversible. It forms a stable inhibitor enzyme complex through covalent bonds and prevents the enzyme from ever working again in something called mechanism-based inhibition. It may kill the organism, and penicillin also works this way, killing something called gram-positive bacteria, which is why penicillin is useful to kill bacterial infections.